Good morning, everyone. My name is Ray, and this is my topic about plastic ingestion by fish in the coastal waters of the Hengchun Peninsula of Taiwan, associated with human activity but no evidence of biomagnification. So my before I start, let me take a short introduction about plastic products. They are very convenient and elastic and maybe low cost, but they also discarded immediately and hard to be decomposed. So, and when they enter the ocean, you will cause the pollution problems. So let me show a short animation to to about this part. When the bottles enter the ocean, and here comes the sun, and the heat may change its structure, and then the waves hit it and break it into small pieces. So if the plastic pieces size is between 1 micrometer to 5 millimeter, and we can call this kind of plastic microplastic. So plastic pollution is a global threat to the marine environment, and if the fish eat the plastic, and this plastic may stuck in their body and make them feel uncomfortable. Like they may feel, oh, I'm full, but actually they didn't eat anything. So this really affects their health. And also, because the plastic cannot be digested, so the microplastic will stay in the fish body, and then the concentration will become higher across the time. This phenomenon is called bioaccumulation. And also, the plastic cannot be digested, so this microplastic will transfer along the food chain or food web, like this picture show. The higher customer, we think the higher customer has higher concentration of the plastics in their bodies. So this phenomenon is called biomagnification. So here's our purpose. First, there is no research on plastic ingestion of fish in Taiwan before. So we want to quantify the plastic ingestion in fish from Hengchun Peninsula waters and then compare plastic ingestion on some biotic and abiotic factors. So here's about the biotic factors that we concern. The first one is the size. It's about the magnitude of the fish body. And next is traffic level. Like this ecological pyramid show, if the location of these animals is lower, and they have lower traffic level, and if the location is higher or on the top place, they have higher traffic level. And the last one is family. It's about the type of the fish. Next, about the abiotic factors. The first is habitat. It's about the location where the fish living. And next is human activity. So here's our research questions. First, do fish in Hengchun Peninsula eat plastics? And how much plastic do they eat? And what factors would influence eating plastic? And then can we see the biomagnification on fish near the Hengchun Peninsula? So to answer our question, we set up some hypotheses. The first is about the family. We thought that how much plastic debris inside the fish would be different among various taxonomic groups. Like different fish contain different numbers of the plastics. And next, about the habitat. We thought that the reef fish will consume more plastic in their body than the pelagic fish because they live closer to the land. The third, about the human activity. We thought that the fish which live in the high human activity area will get more plastic than lower human activity area fish. And the fourth hypothesis, about the size. We thought that the large size fish will contain more plastic in their body than small size fish. This is due to the bioaccumulation. And the last one, about the traffic level. We thought that higher traffic level fish, like the sharks or tunas or others, will consume more plastic than the lower traffic level fish. like the reef fish or others. This is because of the biomagnification. So here's our sampling location, and we set up 7 sites and collect 170 fish samples, and there are 39 species and 18 families. And we can see at this map. We can find that the west coast has high human activity, like more building or others. That is very obvious, and the east coast is low human activity. Like the most of the area is covered by green plants. So, and then where do we find the microplastic? We observe the stomach and the intestine of the fish, and then we put these 2 organs into the potassium hydroxide and dissolve the tissue, and we can get the plastic debris, and then we can use the microscope to do the observation. So after the observation, we do the statistical analysis. So and found that do these factors correlate with microplastic ingestion or not? So after our observation, we found that among 107 samples, about 95% have plastic in their gastrointestinal tract, and all of them were microplastic. And we can look at these 2 charts. The left chart shows that most of the type of the plastics is fiber. It's very thin and looks like a thread. And the right chart shows that the fiber color, most of the fiber color are blue, and followed by white and transparent. So how do type and color of the plastic means? About the type, we speculate the sources of microplastic are textile industries or laundry. And about the color, we suggested that the fish in this area may selectively ingest blue microplastic. So after the research, we found that the results reject these 4 hypotheses. The only one that we found have association is human activity. So because our primary target is the biomagnification, so we focus on the traffic level part. We use the Spearman rank correlation and calculate the p-value is equal to 0.849. And if the p-value is quite higher than 0.05, there is no significant correlation between traffic level and the numbers of the plastic that fish eat. So we can say that there is not enough evidence of biomagnification. And then we compare the amount of the ingested plastics debris by fish in different areas. Like this picture show, the x-axis is about the size we set 3 sites, and then the y-axis is about the number of ingested plastics. So we can see that there's significant difference between West Coast and East Coast, or South Coast and East Coast. And this result is similar to our hypothesis about high human activity area fish will consume more plastic in their body. So about the discussion part. First, microplastic pollution is everywhere. And then next, fish can, the study also mentioned that the fish can ingest microplastic from other ways, such as the water column or seabed sediments. And next, the plastic may be excreted. This means the plastics leaves the fish body through its anus, like this picture show. So these 2 factors may be the reason that we didn't find the biomagnification in this study. So about the conclusion. First, microplastic ingestion is common in fish around the Hengchun Peninsula. And then next, microplastic microfibers was the dominant type of the plastic of the fish ingestion. And the third, plastic ingestion is associated with human activity. And next, there is no evidence of biomagnification was found. And the last one, we need more research to understand about this topic and this problem. So This is my presentation. Thanks for your listening.